Good morning, everybody. My name is Peterson. Today I'm going to talk about some work I've been doing together with two really excellent REU students, Michaela Siever and Ellen Waddle, trying to understand the causes and also the fitness consequences of shifting flowering phenology in an alpine plant. And so as climate change is progressing, we're seeing that the timing of life cycle events from germination to leafing out to flowering is shifting to earlier in the year. And we're observing this across many species and across the planet. And in fact, I would argue this is really one of the strongest and most consistent biological impacts of climate change that we've seen to date, is this pattern of advancing phenology. And on the surface, we might at first assume that this is a good thing. It's a sign of plasticity sort of doing its job and allowing these species to shift the timing of events to track the timing of suitable climate conditions. And that a consequence of this advanced phenology should be that these species should be able to maintain fitness under these environmental change. However, as phenology advances, lots of other aspects of the environment can become decoupled. So if we think about this hypothetical species beginning to flower earlier in the year, it may experience changes in other abiotic conditions, such as increased exposure to frost, for example. It can become decoupled from the timing of its pollinators, and also experience changes in the co-flowering community, both other individuals of the same species or other co-flowering species that may compete with it for access to pollinators, for example. And so given really the complexity of environmental variables that determine the fitness effects of phenology, we might expect that with advancing phenology we would see reduced fitness overall, but also the potential for novel selective regimes as populations are advancing their phenology. So I set out to test these ideas in this long-lived alpine cushion plant, Silene acaulis here. And this is a great system for understanding how climate change can affect phenology and what that will mean for fitness. Because it's a long-lived species, so individuals can live for hundreds of years. And in long-lived species, changes in phenology may be one of the first impacts of climate change that we observe. But understanding what those changes in phenology actually mean for fitness can be really challenging for long-lived organisms. And also, this species has a really interesting mating system. It's gynodioecious, so individuals are either hermaphroditic, meaning they can produce both seeds and pollen and are capable of self-fertilization, or are females. They don't produce pollen and are completely reliant on receiving outcross pollen from insect pollinators. So for this work, we set out to test sort of three main questions. The first is simply, what is driving the timing of flowering in this species? And then second, how does flowering phenology affect individual reproductive success? And does that selection on flowering time itself change with shifting climate conditions? And then lastly, what might be mediating the selection on flowering time? So to do this in 2016, I began working with REU students at Niwot Ridge in Colorado to monitor the timing of flowering. And the first 2 years of this work have recently been published, but we're continuing to collect these data. And in order to link data on phenology to fitness, we did this work within existing long-term plots that are part of a long-term demography study that tracks the reproductive success of these plants each year. And that was initiated by Dan Doak and Bill Morris. And then to begin to understand what might be mediating selection on phenology, we've done a series of pollinator observations and also pollen supplementation experiments. So this is a map of the 4 sites where we've done this work. You can see that some of them are located on these ridges where the wind really blows a lot of the snow away. There's never very much of a snowpack, and these sites melt out relatively early in the season. Other sites are in these depressions where snow accumulates, there's a deeper snowpack, and they melt out later. And so we are able to take advantage of climate variation both from year to year and also due to topographical variation among sites. And so at each site, we tracked the soil temperature and used that to infer patterns of snow cover and snow melt using multiple eye buttons. And so starting in 2016, we began monitoring flowering time on 628 these plants that are permanently marked and tagged as part of this long-term demography study. We did censuses every 2 to 5 days to count the total number of flowers that were open, so that allowed us to fit flowering curves for each individual, and then also tracked fruit set as the number of fruits produced per flower, and total fruit production as our estimate of reproductive success and fitness. And so one of the strongest patterns that's emerged from this work so far is that flowering time is highly variable among sites and years, and this seems to be largely driven by variation in the timing of snowmelt. And so if we look at flowering time either as the date of first flower or the date of peak flower, we see both of these are really strongly correlated with the timing of snowmelt, which is itself incredibly variable across sites and years. And if we take just sort of a broad picture of reproductive success, we see that patterns of fruit production are really variable within and among sites and years, but there's this slight trend of earliest flowering populations having relatively low fruit production. And so that suggests that there's a lot of plasticity in the timing of flowering in this species in relationship to snowmelt, but that there may be some cost to flowering too early in the year. And one potential explanation could be that the individuals that flower first also have a much higher probability of experiencing frost during their flowering. So I next became interested in what's happening within populations and years. So how does individual variation in flowering phenology translate to individual variation in reproductive success? And so to do that, I looked at flowering time relative to snowmelt. So here this x-axis is the days from snowmelt to the day of first flower for an individual, and the y-axis is the fruit production for that individual. And we found really strong and consistent effects of flowering time on fitness. And in most sites, we saw stabilizing selection, where some intermediate flowering time is maximizing reproductive success. And only in this last site, which is always the latest to melt out in the year, did we see directional selection for earlier flowering relative to snowmelt. And so this suggests that when a plant flowers after snowmelt has big effects on its reproductive success, and that there may be, in most sites, an optimal time to flower after snowmelt. So I became interested in the potential for plasticity to allow populations to track this optimal flowering time under different conditions. So I next looked at the optimal flowering time for each of these 3 sites for which we saw stabilizing selection, and that's shown as these horizontal lines here, and compared that to the realized flowering time that we saw in each site and year. And so you can see that under really early melting conditions, even though plants are flowering much earlier in the year, they're still flowering quite a bit later relative to the timing of snowmelt. So they're farther away from that optimal flowering time in these early melting years. And so this suggests that although we see a lot of plasticity in response to snowmelt, it's not sufficient to allow populations to track this optimal flowering time when there's early snowmelt. But then I became interested in what's driving selection for this optimal flowering time. Why is this intermediate flowering time best? And one potential explanation is that it affects the pollination environment, and we see some evidence for that in our system. So individuals that flower at an intermediate time after snowmelt tend to have the highest synchrony in flowering with their conspecific neighbors. And we know that this is important from some pollinator observations we've done, where we found that having a really high conspecific floral display surrounding you increases both the rate at which pollinators visit you, but also the proportion of visits that pollinators make to Silene versus other heterospecific co-flowering species. And so this suggests that flowering at sort of an intermediate time after snowmelt is really important for being able to flower synchronously and thus attract pollinators. But in order to tie this to the changes we're seeing in reproductive success, we need to understand to what extent might reproduction actually be pollen limited and thus likely to respond strongly to these changes in the pollination environment. And so to get at that, we did a pollen supplementation experiment where we had control open pollinated flowers and other flowers that received supplemental pollen. And we found that for both the number of fruits per flower and the number of seeds per fruit, were much higher for flowers that received supplemental pollen than open pollinated flowers, particularly for female plants, which, if you remember, are not capable of self-fertilization and are totally reliant on receiving outcross pollen. And so this does suggest that reproduction is strongly pollen limited, particularly in females. Okay, so taken together, I think so far we've made some progress in addressing these three questions. So in terms of what drives flowering phenology, we see that it's highly responsive to snowmelt in this species. And so we would expect, with continued climate change, as snowpacks decrease and snowmelt occurs earlier in the year, that we would expect to see flowering of the species occur earlier and earlier in the year. However, there's some real fitness consequences to this advancing phenology. We find that there are costs to flowering either too early or too late relative to snowmelt, but that plasticity is not sufficient for populations to be able to track that optimum under really early melting conditions. And in thinking about what might be mediating selection on flowering phenology, we've begun to find some evidence that there may be a role for pollinators here. So we find that the timing of flowering relative to snowmelt has strong effects on the pollination environment, and that reproduction is pollen limited, particularly in female plants. However, I think the next step is really going to be to understand how pollen limitation itself may change over the course of the flowering season. So we're initiating a new experiment this year to do this pollen supplementation treatment on flowers throughout the flowering season that will allow us to ask, what determines the fate of a particular flower based on when it decides to open. So for example, we might expect that the earliest to open flowers may be strongly pollen limited and potentially also limited by some other factor such as exposure to frost. Whereas perhaps flowers that open late may be less pollen limited and more strongly limited by some other factors such as water. And so I'm excited about this experiment because I think it will allow us to begin to really disentangle how these multiple aspects of the environment drive the fitness consequences of shifting phenology. So with that, I just want to say thank you and acknowledge the many people who have been involved in this work, and then just briefly mention that I'll be starting my lab at the University of Georgia in January. So if you're interested in these types of questions or this type of work, and you're looking for postdoc or PhD opportunities, please get in touch with me. I'd love to chat with you more. Thank you.
You do?
Yes.
It's good to sound.
Were you able to measure selection within your hand in open pollination treatments?
That was sort of the original goal, but we weren't able to get fine enough sampling to be able to really relate it to flowering time. But that is, I think, the next step.
Because you would really expect there's going to be a difference in selection between the hand and open pollinated ones if that's really pollinator mediated.
Yes, exactly. So that's sort of the next step, is to do this for many more flowers.
Do you know if the phenology of the pollinators is also tracking these snowmelt changes?
We don't have great information about that right at this site. There's other evidence sort of throughout the Rocky Mountains that pollinator availability seems to be sort of tracking the variation in snowmelt. So I think that my sense so far is that has more to do with being able to flower synchronously with conspecifics, because the pollinators seem to be there, but whether they're visiting Silene versus other co-flowering species seems to be dependent on sort of the local floral display in any